Good morning, everyone. Thank you for joining us at the Discovery Life Sciences Breakfast Symposium. I really appreciate you getting up at 7 in the morning on the last day of the conference. So thank you so much for joining us. We got a great speaker lined up this morning, but before we go there, just wanted to take a few minutes to introduce Discovery Life Sciences to those of you that are not familiar with our company. So Discovery Life Sciences is really a leader in the biospecimen and specialty lab services area. We do a lot of primary cells, blood, hepatocytes, other primary cell types, and then we also run a lot of services around those modalities as well as others supporting preclinical and clinical work. Most relevant for the ISSX conference is our preclinical business, which houses the Gentest brand from Legacy Corning. So all the products that Corning used to offer in this brand are now available under Discovery Life Sciences. That includes primary hepatocytes, liver microsomes, supersomes, which are our recombinant enzymes, as well as transporter products. So, if you need anything, we are still here today at booth 306, so please come and visit us. Without any delay, I will introduce Julius Judd, who is our guest speaker today. Julius is currently the head of High-Throughput Screening and NGS at CoroBio in the Boston area. But before Julius ended up at Coro, he has a very interesting trajectory with lots of publications and patents as well. So Julius got his PhD from Cornell, where he was focused on transcriptional regulation. And after getting his PhD at Cornell, he decided to move over to the dark side, like a lot of us. And he joined Tessera, where he was instrumental in developing new genome editing technologies for therapies in patients. And after being at Tessera, Julius then joined CoroBio, where he is now heading up some of the discovery work for Coro. So today, Julius will be sharing a little bit about the work that he's doing at Coro in terms of discovering new oligonucleotide modalities for liver disease. So with that, I will turn it over to Julius, and thanks again for joining us.
Hi everyone. Thanks for joining us here this morning. I know it's early on the last day of the conference, but I appreciate your time. It's been a great conference so far, so I'm excited to tell you a little bit about some of our work at CoroBio. So just first, I have to get the disclosures out of the way, and I'm going to give you a brief overview of what CoroBio does, and then I'll talk more specifically about some of the work that my team does. So overall, Coro is an RNA editing therapeutics company. So we try to change the messenger RNA in order to either correct genetic mutations or generate de novo proteins. So we have this modular platform that delivers to multiple cell types, and then we also try to activate biological pathways using learnings from genetics. So our overall corporate strategy follows this 3-2-1 model, where we're aiming to have 3 programs in the clinic, 2 tissues that we target using 1 editing platform. So we currently have Coro 110, which is a therapeutic for alpha-1 antitrypsin disorder, AATD, which I'll tell you a little bit about shortly. That's our first program that's in the clinic with a liver delivery modality. And we have a second program that is approaching the clinic for next year, which we haven't disclosed yet, but that's also liver delivery. It differs from our first program in that it leverages activation of the biological pathway rather than correcting a disease mutation. And then we have a third program where we're focusing on targeting tissues outside of the liver. And the key to all of this is that we have this editing technology platform that we continue to iterate upon with the goal of improving potency and delivery over time. And so some of the basics about how our technology works. At its core, what we do is leverage ADAR, which is an endogenous human enzyme that's expressed in all human cells. The enzymatic activity of ADAR is that it is an adenosine deaminase, and it targets double-stranded RNAs. So you can sort of think of it as an RNA-guided adenosine deaminase. And so what we can do here is we can deliver an oligo that guides ADAR to the specific target that we want to edit. So if you have DNA with the disease-causing mutation, you can then leverage non-viral intracellular delivery of an oligo, which guides ADAR to that disease-causing mutation RNA, and then ADAR then deaminates a specific adenosine into an inosine, which is then read as a G by the translation machinery. So by doing this, you can change A's to G's. You can make A to G edits, essentially, in a highly specific way. And so this enables a broad range of disease areas to be addressed using this technology. So you start with, keep pressing the wrong button. You start with your DNA, which there's multiple different places which ADAR can act here. So the first would be to modify gene expression, which isn't the focus of CoroBio. But then this pre-mRNA is then processed into mature mRNA, and this is where our current focus at Coro is editing mRNA, which is then translated into protein. So there are two main things that we can do with this technology. The first being we can repair a G to A mutation with our A to G editing, to sort of revert the protein back to the non-disease variant, or we can generate a de novo protein with preferred properties. We can alter 12 amino acid sequences. So we're limited by the fact that we can only make A to I edits, so we can't edit all 20 amino acids, but we can change quite a few of them. And so our approach we call OPERA, for oligonucleotide promoted editing of RNA. And we sort of think about this in four main categories. So we start with expertise in AR biology, where we have a focused kind of research group that looks at the biology and structure and properties of the AR enzyme itself. And we try to use these learnings to guide development of more efficient oligonucleotide therapeutics. And then next we have a chemistry group that has very significant experience in oligosynthesis, where all of our test articles are synthesized. We also have a machine learning group where we use AI machine learning to predict how well oligos will perform and design, better therapeutics based on our previous data. And then we leverage known delivery to get at specific indications. So we currently have what we think is a very robust pipeline of different therapeutics. So our first drug that's in the clinic is Cora 110 for alpha-1 antitrypsin, as I mentioned before. This is a therapeutic that leverages ADAR to repair a pathogenic variant, and I'll tell you more about this shortly. This uses a LNP with IV delivery and is currently in a phase 1/2A trial with interim data expected later this year that we're all very excited to see. And then we have a second program, which is currently undisclosed, and we're aiming for DC later this year. This is for a rare metabolic disorder. It's a de novo protein to inhibit degradation rather than repairing a pathogenic variant, and it leverages GalNAc for delivery. And then we have a third program for ALS. Our delivery for this is currently undisclosed, but it's a de novo protein to overcome loss of function and gain of function. And then we have this program for subsets of pain, NaV1.7. This is again another de novo protein to modulate currents. And then finally we have a collaboration with Novo Nordisk where we have up to two undisclosed targets for cardiometabolic indications. So our approach, as I mentioned before, we call this OPERA for oligonucleotide promoted editing of RNA. And the goal of this is to produce what we call a CORD, or a customized high-fidelity oligonucleotides for RNA deamination. So this, in its most basic form, is a single-stranded antisense oligonucleotide RNA editor that guides the ADAR enzyme to a specific target site to perform adenosine deamination. And these CORDs we design with these principles in mind. So we look for high efficiency of editing, high specificity for the target that we're going after. We have computational efficiency for designing and evaluating these CORDs, and then we leverage chemistry and delivery modalities to maximize our efficacy. We also couple this with structural bio. As I mentioned before, we have an ADAR bio research group that looks at the structure, and we see what we can learn from the structure of ADAR in terms of how it interacts with RNA, and then using these learnings, we develop novel nucleotide chemistries to try to improve our editing efficiency or improve our ADAR recruitment. And you can see here, this is just an example of a iterative process where we're designing new CORDs based on previous learnings, and then I'm showing the editing percentage on the y-axis here. So this is sort of this is the control, this is the parent, and then this is the newly designed oligo based on previous learnings. And you can see the potency increase here is quite striking. We have demonstrated that this editing approach of RNA works in multiple tissue types. So on the left here, I'm showing targeting the liver with a lipid nanoparticle for beta-actin. So this is the editing percentage on the y-axis, and then I have a time course here on the x-axis. You can see our editing is around 70% at the first day and persists out 2 weeks. We also have examples of liver with receptor uptake. So this is the same target, but now GalNAc delivery, day 7, day 14. You can see we have quite good, efficient editing, which persists again out at least 2 weeks. And finally, we have CNS with naked delivery with an IT dosing. Again, same target, and quite reasonable levels of editing here. So now I want to talk about alpha-1 antitrypsin deficiency, which is the disorder that our first in the clinic therapeutic, Cora-110, is for. And we're really hoping that we can deliver a best-in-class candidate for Cora-110 for alpha-1 antitrypsin deficiency. So what is alpha-1 antitrypsin deficiency? It's most commonly caused by a single missense mutation, or G to A, in the SERPINA1 gene. And so patients with the MM genotype of SERPINA1 are healthy patients. They don't have any deficiency here. And the way that this works is that the alpha-1 antitrypsin protein is produced in the liver and is secreted, where it then inhibits neutrophil elastase in the lungs. So in healthy patients, you have normal levels of secreted AAT, which then produces normal levels of neutrophil elastase inhibition in the lungs, and the liver and the lungs are healthy. But in patients with the ZZ genotype, which is two sort of defective copies of the alpha-1 antitrypsin gene, the patients have reduced levels of AAT secreted, which causes fibrosis in the liver. And then this also, because it's secreted less, you get minimal inhibition of neutrophil elastase in the lung, which damages lung function as well. So these patients typically have fibrotic livers and decreased lung function. And there's about 100,000 individuals in the US with the ZZ genotype. So Cora-110, what it is a CORD, an oligonucleotide RNA editor, that's encapsulated in an LNP, which we license from Genevant. And we leverage IV delivery to get this into the liver, where our CORD plus the endogenous ADAR enzyme then edits the SERPINA1 mRNA to revert the disease mutation. And so with the PIZZ liver, after we've edited the SERPINA1 gene at some percentage, we then get secreted in a combination of the M and the Z alpha-1 antitrypsin. And depending on how much we can edit, we can modulate the levels of healthy alpha-1 antitrypsin that gets secreted to address the lung functionality issues in these patients, and we can also decrease the amount of fibrosis in the liver. And so our aim with Cora-110 is to restore therapeutic alpha-1 antitrypsin levels in ZZ patients. So we're looking for greater than 50% editing, which will provide total AAT levels within the MZ range. So the MZ are heterozygous individuals who have no lifetime risk for lung disease and a low lifetime risk for liver disease. So you can see the three genotypes here, where you have the different, range from a range of 20 to 50 micromolar AAT in healthy individuals. MZ ranges from 12 to 28, and then the ZZ homozygous individuals have very low levels of AAT. And so if we can restore AAT levels to the MZ range, we know from naturally occurring MZ patients that there's no lifetime risk for lung disease and a very low lifetime risk for liver disease. So Cora's goal of 50% editing using Cora-110 has the potential to provide benefit in both lung and liver disease for the homozygous ZZ genotype individuals. Okay, so now I'm going to tell you about our CORD discovery cycle, sort of how all of this works under the hood. So at its base, what we do is we leverage an iterative screening cycle with the goal of generating rapid improvements in editing efficiency. So I'm showing you an example here from one of our programs where we were able to increase the editing efficiency at the higher concentrations by a factor of 2.5 in just 9 months. And so you can see I'm showing like concentration and editing percentage here. And so these iterative design cycles start over here on the left with a parent oligonucleotide RNA editor, and then they undergo they go through our Opera platform, which we leverage all of these parameters. So operational efficiency, we have teams designed, like nimble teams designed to rapidly design oligos, synthesize oligos, and test oligos in relevant tissue types. And then we capture the data and we leverage machine learning to speed up our design process and go through these iterative design cycles. And finally, we are set up in such a way that we use cross-target and tissue learnings to generate even more rapid and potent improvements in editing efficiency. And so the end result of the cycle is an improved oligonucleotide RNA editor, which then feeds back into the cycle and becomes the new parent for the next iteration. So we go through many of these iterations to try to maximize our editing efficiencies. So typically this starts, you can think of this as a kind of classic design, build, test, learn cycle. And so this process starts with either rational design or machine learning. So we have a combination of really skilled people designing these chords, and then we also have computationally designed machine learning chords. And then this goes into the build cycle where we perform oligosynthesis and compound management to make sure that everything is synthesized properly, QC'd, diluted, etc. And then where I come in is the test component down here. So I lead the high-throughput screening and NGS/dPCR groups. So this involves delivering the cord to cells and then measuring the editing efficiency. And then from that, those data sets feed into our data science group where we leverage combined learnings across indications to figure out what changes produce the best chords. And so these changes can be anything from primary sequence of the oligo to modifications, chemotype. We have highly complex oligonucleotides and therapeutics. And so we aim to keep this whole cycle to 4 to 6 weeks so that we can just keep rapidly iterating and generate these improvements in editing efficiency in the timelines that I mentioned before. And so in order to keep this to 4 to 6 weeks, my part of the process, we aim to keep to 2 weeks. So we have this highly automated kind of closed loop system for testing oligonucleotide therapeutics in cells. We typically use primary cells for reasons that I'll get into shortly. But this can be, so we'll plate cells and then we'll deliver to those cells either by transfection or free uptake or other delivery modalities. Then it goes through an RNA extraction and cDNA conversion step, which is highly automated, and I'll show you some of our technology for that shortly. I think it's pretty cool. And then we have a readout, which is typically dPCR or NGS, amplicon sequencing, that allows us to measure the efficiency of editing by comparing edited transcript to unedited transcript. And then analysis. We have these very robust data science pipelines that automate much of the analysis and ensure that everything is reproducible. So in order to meet our 2-week turnaround time goal, we implemented this state-of-the-art automation platform where we have a fully integrated takedown and NGS prep card that allows for simultaneous walk-away processing of up to 4 384-well plates. And this process we've tested quite extensively, and we found that it's highly correlated to manual results across multiple targets. You can see here the automated on the x-axis and the manual process on the y-axis for two different targets. And these are all editing percentages, so you can see they're quite well correlated. So I'm just going to show you a quick video of our robotics platform in action, because I think it's pretty interesting. So this is just the cDNA conversion part, and I'll kind of narrate what's happening here. So this robot arm is taking a 384-well plate and putting it into a 4C Lyconic incubator. Here we have it going to a plate peeler to take the seal off the plate. It comes out an unsealed 384-well plate. Gets picked up again. So this plate has RNA in it at this point. So we've already done the whole transfection and RNA extraction process. And we're going to a Certis flex here to dispense small volumes into the plate. This dispense step is DNase to eliminate eliminate genomic DNA from the RNA samples. It'll then go to a plate sealer where it gets sealed, and then to a thermocycler for incubation. So there's a quick spin step too. So this platform really is what enables us to process the volume of samples that we need to generate these really large data sets of oligonucleotide editing values that we need for our machine learning and rational design approaches to maximizing cord editing efficiency. This lets us have one technician process 8 to 12 384-well plates a week if necessary in terms of plating cells, transfecting oligos, taking down, extracting RNA, and then converting to cDNA and handing off for next-gen sequencing. One other thing that we've found is that lot validation of primary cells is absolutely essential. Different lots of primary cells, so most of these workflows that we have are in primary cells, but different lots of primary cells, depending on how healthy and how well they attach, can generate sort of very different editing values. As you can see here, like the higher concentration, this is 4 different lots of PHH. And as you can see here, at the higher levels of concentration, it's not dramatically different. But then as the concentration decreases here, lot 1 and 2 have much higher editing values than lot 3 and 4. And this is generally what we see when a lot of cells isn't quite as healthy, right? It doesn't plate as well, doesn't stay on the plate as well. You have a dropping off efficiency. And we have found that typically we have very good results with the GenTest hepatocytes from discovery in terms of lot-to-lot consistency and attachment and health of the cells. And so this is a question that I get sometimes, like, why do we screen primary cells? Why don't you just, if we're just looking for efficiency on an oligonucleotide therapeutic from an editing perspective, why do we need to do this in primary cells instead of using a cell model? So what we've found is that editing results are highly cell model dependent. So here I'm showing a more recent experiment where we had 2 different cellular models, and then I'm plotting editing efficiency in percentage, and these are just different compounds. And so you can really see that compounds that are optimized for maximal editing, like in cell type B, these are all good editors, but they're quite poor editors in cell type A. And these are reasonable editors in cell model A, but they're quite poor in cell model B, right? So these results do not translate uniformly across cell models. So if we spend a lot of time optimizing oligos to edit RNA in one cell model, and then we try to go to primary cells or an in vivo model, we may not have good results. And furthermore, when we look at cancer cell editing, you can see here, so this is a cancer cell line on the X compared to editing in primary hepatocytes on the Y. And you can see like there is some correlation here, and the editing is generally lower in hepatocytes than it is in the primary cell or the cancer cell line. And while there's a reasonable correlation in editing values, there's some editors which are way off the sort of the correlation line. And importantly, when we rank order these, so this is one method of selecting which oligos are performing the best is by rank ordering them by editing value. When we rank order these based on the cancer cell line data, we see that this oligo that was 19th in cancer cells is the second best performing oligo in hepatocytes. And it's like the best performing oligo was in the 5th position. So if we were to, assign an arbitrary cutoff based on a primary screen in a cancer cell line and only look at the top 5 or the top 10 oligos, we're not going to be selecting the oligos which are going to perform the best in hepatocytes. And we're going to be missing some that would be quite potent editors. And so to delve into this even a bit more deeply, we've actually looked at this based on different classes of oligonucleotide modifications. And so here again, I'm showing a cell model A editing percentage, and then primary hepatocytes editing percentage. And then the color coding here is different classes of modifications on our oligonucleotide therapeutics. And so what I want to draw your attention to here is modification class A, where you can see that this modification class clusters up here where it actually boosts efficiency quite a bit in primary hepatocytes, but not in cancer cells. And then there are other classes where it is pretty well correlated. So it's not this is the biggest issue with this is that it's not uniform, right? So depending on how you're modifying the oligo and what the exact chemotypes you're working with are, you might have good translatability from a cancer cell model or a different cell model, or you might not. And in this case, we actually found that this modification class A in this specific model actually decreases editing efficiency in the model while it increases it in primary hepatocytes. So you can see here, I'm showing here editing percentage by concentration, and I have the unmodified oligo in gray here in the cell model, and then the modified oligo here in the cell model, and then the same for hepatocytes. And you can see you actually get a decrease in efficiency in the cell model while you get an increase in efficiency in the hepatocytes. So all of these things together are why we choose to screen in primary hepatocytes, because we really want to optimize our efficiencies in the most therapeutically relevant cell type that we can, so that when we go into in vivo models or into the clinic, we have a well-performing oligo from the start that isn't optimized for the incorrect cell type. So in summary, today I've told you about Korro's Opera platform, where we utilize CORD technology to edit RNA using endogenous ADAR protein. CORD discovery relies on iterative screening cycles to generate rapid improvement in editing efficiencies. And we have found that compounds optimized for maximum editing in one cell model do not necessarily perform well in other cell models. Editing in cancer cell lines does not uniformly translate to primary cells, and this is particularly obvious for certain classes of modifications which improve editing in primary cells but don't improve it in cell models. And this primary cell-based screening paradigm that we have really ensures maximal editing efficiency of our core technology in therapeutically relevant tissues while maintaining the shortest possible development timeline.